So we are back from the break, and request you everybody to go back to their seats, and hopefully you had a good break. For the next talk, we have Dr. K.V. Venkatesh. He's going to talk on complexity in living systems. So whenever one thinks of complexity, like what we generally think of thinking electrons in case of social context, or we think about living cells, invariably the mention of living systems as complex systems comes forth. The spectrum of complexity is very special. For that, the Earth is the only known place which is harboring the life. How the life works, in particular, for the in an applied sense, how the human physiology works, has its own important applications. Today we have Dr. Venkatesh to share his insights on this topic. He is professor and head of department at Chemical Engineering at IIT Bombay. He is a pioneer in the areas of systems biology, network analysis and modeling, synthetic biology, and metabolic and regulatory networks research in India. With more than a decade of experience in this particular research areas, he has contributed significantly to research in the areas of quantification of biological networks, including genetic signaling and metabolic pathways, metabolic engineering, and his group has developed steady-state gene expression simulator methods using elementary modes to quantify phenotype space to complete whole body metabolic model for humans. He is a recipient of several awards, including INSA Young Scientist Award to being an international judge for internationally genetically engineered machines, iGEM 2009 by MIT USA. His talk will focus upon bottom-up models of human physiology from cell to human and how complexity manifests itself in such a system, and to explore relationship between lifestyle and emergent conditions such as type 2 diabetes. Over to you, Dr. Venkatesh.
Thanks a lot, Ashil, for that introduction and inviting me for this meet. I think it was interesting that we started with physics, then maths, and then we've come to engineering now. Looking at the same problem of biological systems, but different sort of perspective. So I'll start with, for the audience who know knowledge in biology, that, I think the previous speaker also sort of indicated that you have the central dogma where you have genome, you go to transcriptome, which is decoded, the gene is decoded, and then you have a protein. The protein carries out a reaction, so it's a metabolome, and then you have the phenotype, which is the characteristics that is visible. So this is a hierarchical connection, so we all have genes, it gets decoded, and then proteins are there, and then the reaction does not happen spontaneously under ambient conditions, so you need a catalyst, and then the catalyst is this enzyme or protein, and then a set of reactions manifest itself into a phenotype. So the presence of genome does not mean that you have the phenotype, so it requires a specific state and connection to yield a particular phenotype. Okay. So for example, you have the luciferase gene, okay, you can decode it, gives RNA, you have a luciferase enzyme, so it essentially, the phosphorus it releases using ATP, it glows, right, and then that's why your firefly glows. The phenotype of a firefly is that it glows in the night. So why it is able to do that? Because it has got a reaction, it has got an enzyme, it's decoded, and in a coded form it's a gene. So this hierarchical connection makes a firefly glow. So in 1983, what was done was this luciferase gene was transferred into a tobacco plant. The gene was decoded in the tobacco plant. The enzyme was catalyzed in the tobacco plant. The reaction was carried out in the tobacco plant. Then what do you see? The tobacco plant glows, right? So this is, humans have created a plant system where a biological system which exists in nature, so that phenotypic property has been transferred to a plant system. Okay? So this is engineering, right? So we've engineered a new system which starts glowing, okay, because we understand this hierarchical connection, and then we can transfer this to another system. But in a real sense, this is not engineering. because this was purely done without any quantification. Okay, it was driven in a protocol. It was done in a protocol, so something like a recipe, right? Something like cooking, right? So we cook, that we have a steps to follow, we do that, and then you have an item, right? So similarly, so you have a property of glowing. So this was purely done based on a recipe. That you take a gene, insert it. So a lot of trials were done, empirical trials, before we got this system working. But engineers don't work this way, right? So engineers, we want quantification. So we want to know components, how the component works. I want to bring these things together and then try to see, how a property is. So we design things in precisely in a quantitative manner. But if you look at biology, historically biology has been a descriptional science. Okay, so most of the engineers would say that, okay, I'll stop studying biology in 10th because there's a lot of description. mRNA, xylem, phylum, we'll have to add so many things. So most of the engineers who are here would have stopped studying biology. Okay, that is essentially because it is a descriptional science. That's the perception that we have. Molecular biology, okay, in the last 3 decades, has essentially given this molecular level description. So the modern biology is also description, but the description is at the molecular level. Okay, that's how we come up with networks. That we have been discussing in the morning. So the networks came about because we looked at a directionist approach. We broke up the cells and wanted to see which components interact. So what this results in is we have a genetic, signaling, and metabolic networks. And this is the result of the reductionist approach of the molecular biology. Bioinformatics adds more this information, okay, by comparing different systems and say that, okay, if I see a connection here, do I see the same connection in some other system? Okay, the pattern matching, and it does more connections. Right? So this results in a highly interconnected network. And this is what we have been saying, that the complexity comes about because of this such highly interconnected networks. But as I was indicating, engineering systems are quantified. Okay, it is quantified to the level that we can design them, we can optimize them, and we can optimally operate. Right? So we have now the capacity to send a satellite and to go around the sun. Okay, how are we able to do it? Because we know the physics, okay, we know the complete design, we understand how the systems operate, all the quantification is known, I know how to escape gravity, right? So I can design a system. So that's why it is completely quantified. Biology is also now evolving to be highly quantitative science. There are 2 new methodologies which have come that I can now quantify different components. So now I can apply the principle of system science, okay, which the engineers are familiar with, and apply it to biology. Okay, so this is what is called as a systems biology. So we are trying to apply all the principles that we use in designing and understanding engineered system to biology. So as an engineer, okay, I would design something, I would operate, I'll control it, okay, I'll see at some faults, the system may have faults, then I will go and redesign. So I go through this process, and this is what gives rise to evolution of new systems. So if you look at the aircraft, which was designed by Wright brothers, and the aircraft that you see today, they're completely different, right? But the phenotype is the same, okay? The phenotype is that I need a flight which is stable, okay? Something has to fly, and it has to be stable. That's the phenotype. But the design that the Wright brothers have, and the Airbus and all that now you see in the modern world, okay, are completely different because they evolved. Right? The system has evolved. Natural system also goes through the same process. Okay, it evolves over a longer time scale, but they also evolve. So for that evolution, new designs have to come. Okay, so the life principle may be same in a bacteria, for example. In humans, similar things operate. Okay, lactic acid metabolism happens in bacteria, lactic acid metabolism happens in us, right? So it is conserved. So that part of the design remains. But on top of that, new designs have been built, okay? And that's what is the evolution process. So new designs have been added. So we go through this process of designing something, we operate it, we control, okay? We look at faults. If this fault is not taken care of, what happens to the system? Okay, it gets eliminated, okay? So it gets eliminated because it's not able to adjust to the perturbations that exist, like dinosaurs, right? Dinosaurs got extinct because it could not evolve into new designs, okay? So it gets eliminated. So this evolution process becomes very important. So if I was giving you the example, aircraft. So if you look at a modern aircraft, it has about 1250 computers, hundreds of feedback loops, and we now know that the feedback loops are very important. Okay? Millions of components which are present. Design manual is available, okay, because we have constructed it. It is a bottom-up design because humans have built the system. And pretty much only takeoff requires manual effort. So it's almost a system operating by itself. But you'll see that there's a lot of calculations that is going on, a lot of quantification that is going on to keep the system stable or operational. And that is why you have these 1250 computers. There are a lot of sensors on the top of the wings, which is fed into the computers, and then the computer is helping take decisions. If you look at bacteria, which is a gut microbiome, okay, a gut bacteria which is there in the human gut, okay, it has about 4,400 genes. A lot of connectivity between genes, mRNA, proteins, a lot of connections. A modern aircraft has hundreds of feedback loops. Bacteria has thousands of feedback loops. Okay? So a simple bacteria is more complex than a modern aircraft. Okay? That's what it implies. Because already the connectivities are far greater. Okay? Let alone the human system, just a bacteria itself has thousands of feedback loops. It appears that there is no design principle because I don't have a manual for bacteria. Because I have not designed bacteria. The bacteria exist in nature. Right? So if I have satellite or car, computers, I can have a manual because I have built it. Okay? But these are top-down systems. These systems exist in nature. Okay? They've evolved. Okay? Like what he was indicating in the morning. So randomly these things came and it evolved. Nobody designed them, but it evolved. So these are top-down approach. These systems exist. So I don't have manuals for them. Okay, there is no design principle. It appears that bacteria does not do any competition, right? So there is no competition, right? So the control and sensing actually each time two proteins come together or a protein interacts with the DNA, it is helping the bacteria to design, to take a decision. Okay, actually calculations are happening. Bacteria shows a phenotype, what is called as chemotaxis. Okay, so chemotaxis is that this bacteria can go from a lower concentration of glucose to a higher concentration of glucose. So it can traverse, right? So bacteria is able to do this. So this is a very engineering thing to do, right? If supposing I want to construct a robot which goes from a lower concentration of glucose to higher concentration of glucose, if I want to build a robot or a system which is capable of doing this, what do you need? So I want something to go from a lower concentration of glucose to a higher concentration of glucose. So what do you need for this to happen? Sensor. So you need a glucose sensor. Okay. You need a motor. Correct. And you need the sensing component information to go to the motor. Okay. Further, it has to know the concentration of glucose at one point. It should traverse, measure the concentration of glucose again, take the difference, okay, get the gradient. If the gradient is positive, it has to move in that direction. If the gradient is negative, it has to change direction and then move, right? So as an engineer, I can design this. But E. coli is already doing it. So how is it able to do this, right? Because the design exists inside. So now we precisely know how bacteria does this. So that we know the sensor, we know the signaling pathway which sends the signal to the motor. It has a motor. The flagella rotates in clockwise or anti-clockwise direction to decide which way the cell has to move. Okay. We know the memory element, how it is calculating gradient. So we exactly now know. So that means that I'm building manual for E. coli. I'm saying that E. coli is demonstrating a property of traversing from low concentration to high concentration. But now I can write a manual and say how precisely it is doing. Okay? It is just not network description. You need quantification. I need to know exactly what is the binding constant of glucose to a membrane protein which is present on the membrane, the protein which is present. So what is the binding constant? What is the time constants for the signal to go from the membrane to the motor? Okay? What is the energy needed for the motor to drive? Right? So this is what an engineer would try to quantify when he's writing a manual. I have to describe each component, and not only that, I have to rate the component. What is the quantification of that? So it is just not that I have a circuit. I need to know what is the resistance of a circuit, what is this capacitance, what is this rating. I need to know that. Okay. So precisely in living systems, I have to characterize that whole thing. But now E. coli does not have only one phenotype. It is not just chemotaxis that it does. Right? So this is a system which is capable of demonstrating many properties. Okay? It is not that we just walk on straight on two legs. That is one of the phenotype. We do many other things, right? So aeroplane is you have to just stably, it has to take off, stably go and land. That's its phenotype, finished. And that is what we have designed it for. But biological systems, they demonstrate a lot of varied properties depending on the environment, depending on what are the perturbations that exist, okay? And its own survivability, okay? It has to come up with designs. So when I'm writing a manual for E. coli, I have to keep track of each of these phenotypes and then try to see and describe quantitatively the complete description, okay? Not only of the network, but also of the numbers, the quantitative information, right? So that is where the challenge comes in, because these are all highly complex systems, okay? So how do I independently measure each of these components? Okay, I have a protein network, okay, and I want to know at what time this system gets phosphorylated, right? There are time constants involved, there are reactions involved, interactions involved, right? So I need to lay down each of these components for a particular state. Okay? So in engineering term, there is what is called as a state estimation. So for a particular performance, I need to have a state estimation. Can I do that for biological systems given the quantitative information? Right. So now there is possibilities. Okay, this is another way of understanding complex systems. That given the parameters or even incomplete measurements, how can I rationalize using state estimation what is the state that it exists in? Right. So can I rationalize that? So I give this example to my students. We have had many examples to quantify what is complexity. Right? So I think we have heard this in the talks in the morning as to what this complexity is. Right? So I give this example of defining what is complexity. Okay? So I take a blue ball and a red ball. I randomly pick a ball. Okay? Supposing it is a blue ball. So what is the probability that it is blue? 50%. Okay? I will slightly change the experiment. What I will do is every time I randomly pick a ball, I will replace that particular color ball in the next step. Okay. So if I randomly pick a blue ball, in the next step I will have 2 blue balls and 1 red ball. Okay. So this is the new experiment I'm doing. So now if I do this 50,000 times, now I ask you the question, what is the probability that it is blue or red? What will that be? No, it need not be first ball. It also depends on the history, essentially. Okay. So because I have created a nonlinear, step, okay, in the process, now I don't know what will be the probability. I start with 50-50, but I can go here, I can go here, I can go anywhere I want, right? So what is it telling me? Okay, it's essentially telling me that there are multiple outcomes that are possible. And it depends on the history. Okay? So a complex system can go to many states. Okay? It can go to many steady states. But the steady state it goes to is dependent on what? It is dependent on the history. So we are all Indians. If I take the genome of all of us, the variability will be less than 0.001%. Okay? That will be the variability. Okay? And we all start as a single cell. Okay, we all start as a single cell. So the variability at the system level, when we all start as a single cell with half the chromosomes from father, half the chromosomes from mother, okay, and we all start as a single cell, the system is the same. There is no differentiation. It is less than 0.0001% difference. Okay? But are we in the same state? We are all different. Okay? So that means that we have ended up in, we all started out as a similar state, in almost same cell. Right? But we end up in different states. And that is because of the complexity. Right? We have our own history, our own perturbations that we encounter. Okay? Both the system, okay, the genetic differences, and not only that, the environment. Okay? Somebody is malnourished. By chance, that is what he undergoes. Somebody has a polio virus infected when he is infant. Okay, his whole phenotype changes. Right? So that's a perturbation that he goes through, and that will take him to completely new steady state. So we are going in a healthy state, perturbation, suddenly we become diabetic, right? Or we go into cancer state. Right? So these are all different steady states that the system can go to. Okay? And this drives because of our history. Okay, what are we experiencing? Okay. And the steady state that we go to. Okay, there's a very good program on BBC Horizon, and I don't know how many of you have heard. It's a very interesting scientific documentaries that the BBC produces. So obesity. So they were trying to understand how obesity occurs. Okay, in US. So they found out that there are people who are dieting, who are exercising, who are very fit, but they're all they're becoming obese, right? And they pointed it out to viral infection. Just a viral infection changed their complete metabolism. They started storing fat in a very high rate, right? So it has nothing to do with their physical activity. It is just that some perturbation, they got a virus into their system. That's it. Okay, their whole metabolism got altered, right? So this is a perturbation. So how it can take you to complex system, can go to a new state and change the complete metabolism. Okay. So these kinds of things can happen because we have multiple states that we can go to. So what are we dealing with? Multiple steady states, depends on history, parametric sensitivity, bifurcation, robustness, adaptability, evolvability. So these are all coming about because of the complexity. Okay, these systems are able to show these kinds of properties. What are we to deal with? Nonlinear dynamics, multiple feedback loops, multiple interaction, cascade structures, feed-forward loops, interaction between modules, timescale separation, which engineers are very familiar with. Okay, so engineers keep dealing with these kinds of things, trying to understand how systems are nonlinear, cascade structures are there. Okay, and there is a timescale separation, something happening in seconds, something happening in hours, something happening in days. So there is a flower which blooms in Kerala once in 12 years. Okay, during the springtime, with an error bar of 2 weeks, 2 to 3 weeks. So in March, in a 3-week period, this flower blooms once in 12 years. Okay, how is the system able to do this? So there has to be a design inside. So if I start writing a manual for this plant, so there will be a time constant. There is some clock inside the system which is keeping track that when that 12 years happen, when those 3 weeks, everything has to come together, and that is when the flower blooms. Okay. So now can I write a manual for this and say what is that clock mechanism? How is that that system is able to precisely say that something is going to happen in 12 years, right? So this is amazing, right? So systems biology tries to build such manuals, trying to understand what is the design which exists to show a particular property. So what is this that we need to do? Right? So you break the components, and we are trying to understand what the component is. So I could not do systems biology 3 decades back, right? Because I didn't even know the components. So the first thing is I need to know which components are interacting, right? So the reductionist approach is the first step. I need to know what those components are. Right? So the radio was not working, so I need to understand each of those components. What does it do? I have to rate it. Okay. Only then I can now try to understand what the radio does. So this is essential. So I cannot escape this. Once I have these components, I can bring it together. Reconstruct, look at the systems level, and now look at components, reconstruct it, look at modeling simulation, do feedback analysis, okay, do Monte Carlo simulation, try to understand what these input outputs are. So all that now I can do, right? Which the engineers typically do. So this is the first thing. So we have to look at an integrative approach. Because the sum of the parts, is not just addition, it is more than that. So I have to now bring these two together and then try to see what are the phenotypic properties that now this integrated network can give rise to. Okay. So what do we do? So if supposing I want to look at this cellular complexity, so I need to do mathematical modeling and computational simulation. Okay, I have to put all these computational tools in analyzing biological, medical, or healthcare data that may exist. So the quantification I was talking about, so the genomics, proteomics, metabolomics, interomics, so all this, there are now tools, LCMS, okay, trying to see which protein is phosphorylated. So now there are tools to actually quantify them at different levels. So that's how the quantification is also growing. Not only the network description, but also the quantification. So we integrate both this information. And now I look at quantitative mechanistic based understanding of the cellular machinery. So now I can bring that onto the platform, okay, and try to understand what this input output would look like. Okay. So what is the complexity that we are talking about? I mean, this has also been indicated in the previous talks. I start with molecules, I go to pathways, I look at cells, I have tissue, and I go at the physiology. So both temporal and physical, spatial, okay, both different dimensions that I need to consider. Right? So this is the complexity that we are talking about, that I need to have a multi-scale model. There are processes which happen in seconds, processes which happen in years and decades, okay, and I have things which happen in nanoscales and then in huge spatial differences. Okay, something happens in liver, how does it affect the brain, right? So these are completely spatially separated. So how do I link this? So this both temporal and spatial separation or the complexity exists in both the scales. So you have external factors, okay, environment, lifestyle, diet, for example. You have a phenotype, how our body responds to these perturbations. You have metabolism, okay, the metabolites and metabolitic enzymes which are present. So these are reactions that take place. And then you have signals, okay, which signaling pathways give, and then you have the state, the genotype. Right? So these are all interconnected in a complex way. So the genotypic response to external factors perturbation and this remaining in a certain state. Okay, so that's what I was talking about, a state estimation. So what are the states that all these parameters exist in for a given phenotype? So that is the challenge. So disease, as we characterize, is a systemic defect. Okay. Even if you have an infection, right? So it is something that is perturbing at the systems level. Sometimes the perturbation is genetic disorders, but it manifests down the line in any of these cases, communication or reaction. It can go to any scale. It could have a metabolic issues, it may be a metabolic problem, but then very quickly you will see that some mutation gets selected because of that perturbation. So the disease may start with some perturbation, but the final manifestation is completely different. It can happen at some other place. Okay. So this is one point. The other is, what is this healthy state? So how do I define a healthy state? So that itself is a challenge. So the healthy state gets defined only by characterizing defects. Right? So we say that something is not working, this is unhealthy. So to make it functional, so what is it the correct thing? This is how we define what health is. Right? So this is another challenge because there is no definition for a normal behavior of a phenotype because we don't even know. Because this system exists, as I told you, there is no manual for these things. Right? So how do I even define what is a normal design? It's very, very difficult. So it starts at that level, trying to even define what is normal. Right? So we don't know. So typically that is done by looking at defects. Okay, I perturb, create a disease and then understand it and then say, okay, so this is the normal definition. This is how it should function. So obviously, this has engineering applications to it. If I'm capable of developing some understanding, okay, of living systems, obviously I can apply it. For example, human physiology, if I'm able to understand, obviously it can be applied for drug discovery. Okay, that it can be try to see system level insights. I can generate hypotheses, how in a disease, what drug that I need to design for, compound validation, disease indication, even definition of a disease. Right? How do I define even what a disease is? Okay, in an individual. So these kinds of things I can now start characterizing and then listing it. So what we have done is we have taken, this is the typical flow for any system level analysis of a living system, that first you start with the data mining. Right? So I need to go and look at literature, so where clinical data is there, where networks are defined, right? Where perturbations have been done, and then the system is characterized. So I start looking, I data mine and get all the data, the omics data, and then I look at transcription. What are the things that is happening? Networks. I build networks based on this data mining. Then I do the modeling of it. Okay. Then I validate through experiments or existing experimental data, feedback, correct it, and go through this process. And then now I can see whether I can do side effect. Okay. If a medicine is put in, what is has it got any side effects? Okay. So I can now start looking at these kinds of things. Can I have a personalized medication? Can I have a model for an individual? So these kinds of questions I can start answering if I build such a platform of characterizing the state estimation, estimation of states in an individual. So what we have been doing, I have another 5 minutes or 10 minutes. So what we have been doing in the last 10 years is trying to build a whole body physiological model. So we looked at modeling genetic and signaling models at the cell level, take it to the tissue, look at organs and then the individual. So we build tissue-specific models, multi-organ interactions, and then the whole body response. So, and we also try to capture the effect of lifestyle. So we look at a lot of theoretical data, physiological data, and then try to build models, on these kinds of things. So we have a metabolic model for the brain, muscle, liver, adipose tissue, no neuronal, just metabolism. And muscle, complete signaling pathway and its metabolism, liver, adipose tissue, gastrointestinal tract, pancreatic beta cell. Okay, so we have built these models. We have connected to all the signaling pathways, the various signaling pathways. Meal simulation. Okay, so if I take carbohydrate, protein, fat, how it gets absorbed into the blood, how those things vary, and then it go to these, it's a modular, goes into these organs, and how those signaling pathways perturb, and what is the state that it goes to. So we connect this and then try to look at effect of exercise, meal, because all this will give hormones, different hormones are released, and these hormones integrate and look at the performance. So for example, somebody exercised for 1 hour, had breakfast, lunch, exercise, dinner. So now I can actually look how blood glucose is going up, what is happening to insulin, what is happening in liver, what is happening in muscle. So we have some close to 1800 variables. I can keep looking dynamically how things are changing. Okay, because these are all dynamic models. So I can look at how a lifestyle effect is going to bring in. So I can do what engineers do, input output. Engineers are very, very familiar with, I want to know if this is there, what is the input and what is the output. Right? So we all do 10th standard experiment. We put voltage and connect it to a resistor, right? So what do you measure as an output? Current, right? Flow of electrons. Input is voltage. So input is voltage, output is current, right? How does the system behave? Linear, Ohm's law, correct? What is the slope of that line? Resistance. Resistance is a system property. Input is voltage, output is flow of current. If you take a meal, your glucose goes up. The rate at which glucose is absorbed in muscle, just like flow of electrons, flow of glucose. What is the input? What is the potential? A hormone, insulin. Okay? So I'm doing the same thing. The 10th standard experiments that you do, instead of that, now I'm trying to understand how in a muscle, insulin, controls glucose, flow of glucose into the muscle. So this is the input-output curve. Obviously it is non-linear, and it gives a bistable response. Right? So insulin is in nanomoles, in very small concentrations, these hormones, right? So, and these are highly noisy. So this noise, you don't want it to transmit to the y-axis output. So you have a filter. Just like frequencies you eliminate, you don't want any other noise to get. So you have filters. So nature has designed these filters, right? So the noise in the input voltage, you don't want it to transmit it to the output. In a linear system, the noise in the input will transmit to the output. So if voltage is fluctuating, current will fluctuate. You don't have control. But in nature, this system, it has eliminated, it has created a filter. Right? If one of the signaling pathway, one of the molecules, the concentration is doubled, what happens? So this PTPase is one of the signaling molecule. If this concentration is doubled, this input-output curve goes to the right. Your body is producing this much insulin. Now what does this do? Off. Right? So to switch this on, you need almost a order of higher amount of insulin. Insulin is not the culprit. Your insulin is normal. The culprit is another signaling molecule. So your input-output has got changed. Okay? It is in the same experiment example that I was giving. If you keep doing that experiment, right, the metal temperature goes up, it gets oxidized, the carbon suit sits on that, so your resistance changes. Okay? So now the same amount of electron will not go, right, because it has got defected. Now you take a sandpaper, you clean it, again it will start flowing. Right? So something similar, now something has gone wrong, your input-output has got shifted to the right. So what is the real treatment? The real treatment is you need to take this back to original input-output. But you don't know. So in one individual it may be PTPase, in another individual it may be something else. There are 25 sensitive points. So each individual, the disease is because of some unique reasons. But you don't know, right? So what is the treatment? They just say, give insulin. Okay? Or control glucose uptake. You control the glucose uptake. Metformin. So this is a drug that is given. So it just forcibly controls glucose going in. Okay, you have not corrected it. You have not corrected the system properly. Only symptomatically you are giving a treatment because you don't understand as to why a cause is in an individual. So now we can start asking such questions because we have an understanding or we have started building a manual how this works. So we are noting down what this does physiologically. So we can look at many of such perturbation, reduction in blood flow, ischemia, insulin resistance, hypoxia, when oxygen demand is gone, okay? And you can see everywhere metabolically there's a lot of perturbation. Look at cancer, right? It is so stable. It is like a healthy cell. Right? So through NADH, oxidative stress and energy status, you can say, you'll immediately see inflammation, some pain, there are indicators. But for Warburg effect, right, cancer, the metabolism is highly stable. That's why it's very difficult to identify in the early stage, when a cancer occurs, because metabolically there are no indicators. There are no indicators metabolically that, how this happens. So there are many examples. So we look at, signaling pathways, description of how diet changes, how in cholesterol metabolism, so purely like a plant, right? So we actually indicate, what is happening in blood, what is happening in liver. Okay, so we give description in terms of the performance of each of these behaviors. Okay. So I'll just, want of time, just go to stop. I'll just give you one feedback here that one of the studies that we did on cholesterol, the most sensitive point was this intestinal bile salts excreting the cholesterol out. Okay. It was nothing to do with diet. So whenever you see an advertisement that this is zero cholesterol, that does not mean anything. Okay. The cholesterol in the food will not do anything to you. Okay. Your bile, okay, is it functioning properly? That is what is the key thing. If your bile is working properly, so it will throw out the cholesterol. So there was a recent study by Harvard Medical School where they gave 1600 mg of cholesterol every day. Okay. Steady state change, net change, 2 points. From 170, the cholesterol went to 172. Okay, so this modeling, 8 years back we had told the same thing, that food is not going to do anything. Okay, it is the cholesterol, it is the bile salts. What is the medicine that is given? Statins, right? So which decreases the synthesis in liver. So that's also a wrong point. So wherever cholesterol is high, they say, okay, you take statin. That is also a wrong medicine. Okay, it's not a holistic medicine. It is the bile. And luckily, I was just looking at it, and surprisingly, the Chinese medicine and Ayurveda actually target here for high triglycerides. Right? So they say that the bile activity has to be increased if you want to control the triglycerides. Right? So our old medicine is probably at a system level and already understood. Okay? So that's the reason why they ask you to drink kokum, they ask you to take tamarind. In South India, for everything we put tamarind. Tamarind has a natural hydrocitric acid, okay, which actually activates bile. So whenever you are putting tamarind or kokum in your food, you are essentially saying that you activate your bile. Okay. So this is a natural way of doing the same thing. All our system level understanding is giving you, okay, what probably our forefathers had sort of indicated purely as a sum system, their own understanding, okay, in their understanding. So I'll stop with this, okay, and then tell you that, quantification is essential. Trying to come up with design principles for trying to relate input-output, okay, the way the engineers would look like, would look at, okay. So these are some of the things that we have been doing at the whole body metabolic level. So if there are 2 minutes, I can show you a demo that we have built for managing diabetes, okay, with these kinds of understanding. How diet, medication, and the glucose dynamics can be used to so supposing I give somebody with 45 years, say male, fasting glucose is 80, okay, postprandial is 120, okay. Somebody weight is 65, 170 centimeters. Just look at the current status, number of days. Somebody takes carbohydrate, fat in their diet, no medication, takes 2400, lifestyle is active. I can now simulate, right? So it's going to tell you so we predict what is the glucose dynamics that he will have. So we have taken from KM Hospital, 1000 patients, what is a healthy state and what is a diabetic state, okay? So this green one gives you an average for Indians, what is the healthy glucose dynamics. This red is telling you on an average on an Indian population, what is a diabetic glucose dynamics is. So if now I predict for an individual, okay, by characterizing parameters, if some individual puts his data, okay, this is the diet he's taking, this is the physical activity, this is his glucose dynamics. So we can individualize now and get a parameter space for each individual, and now I can predict, okay. So now this will tell you what is the extent. So predicted diabetic risk is 43%. It means that in the next 3 to 5 years, there's a 43% chance that you may get diabetic, because this is for Indian population. And I'm now predicting what would be the dynamics given these inputs. Okay. So what would be the future diabetic risk? So if supposing I change this parameter, somebody has got 130 and 240. Obviously this is diabetic. Simulate. So now you see that it has become diabetic. So you are already diabetic because it's gone above the red line. So now I can say, supposing this is specific only to metformin. Supposing I say that he takes 200 mg per day of metformin. Sorry, days I have to take, number of days. Say for 100 days. It came down little, but no control because his diabetes is very high. So 200 mg will not suffice. So supposing he takes 750 per day. So it came down little. Right? So now you can see the effect of medication, effect of diet, okay, with physical activity, how they can balance all this and they can control diabetes, for example. And behind, our model is running. This is not AI. Okay, so this is a platform which is not giving you just input output. Because of the complexity, okay, input output, AI, as far as I am concerned, is something like a glorified fitting, right? So it's a black box. So I don't know what is happening in the middle. It's an input, output, I just correlate, okay, through AI, MI, or regression, whatever you can use it. Right? But we are not going that way. So we actually want to capture the mechanistic detail, what is actually happening in between, okay? So with that, we try to connect the input output. Okay. So with that, thank you.
It looks like moving out of silos and getting into systems physiology, which will revolutionize the way medicine has been practiced. So now I request Dr. Neeraj Gupta from Ayuka to felicitate Dr. Venkatesh.